Howdy, welcome to my presentation. Today I will be discussing how neuropeptides allostatin AMASTs and tachykinin AMTRPs are responsible for the aggressive behavior of the Africanized honeybee Apis mellifera. Alright, the common behavior of the honeybee includes colony defense, foraging for various resources, thermoregulation in the form of shivering or fanning, swarming for the means to reproduce, and a particularly established eusocial social structure. Now that we have established a basic background of Apis mellifera, I would like to talk about its hybrid form. Africanized honeybees are not the same as African honeybees. Africanized are hybrid species or the offspring of the Western/European honeybee and the African honeybee, which is native to Central and South Africa. The African honeybee is also known as Apis mellifera scutellata, and it spread west in the late 1950s to the Americas and bred with Western European honeybees, and by the 90s created a rather aggressive and invasive species also known as the Africanized honeybee. I was curious as to exactly what makes Africanized honeybees so aggressive, and I did some research on the components. How do neuropeptides allostatin AMASTs and tachykinin AMTRPs affect the behavior in Africanized Apis mellifera? I focused my finding on a research paper that used MALDI imaging analysis of neuropeptides in Africanized honeybees to study what neuropeptides are responsible for this aggressive behavior presented in these honeybees. The first The study first had Africanized honeybees placed in an environment with leather targets. The bees that aggressively stung the targets were placed in liquid nitrogen and frozen. The heads of the bees were then frozen and sliced for the MALDI spectral imaging analysis. The process is shown in the figure on the right. The precursor proteins found were turned into the mature forms of AMASTA 59 through 76, AMASTA 69 through 76, AMTRP 88 through 96, and AMTRP 254 to 262. The data presented in the figure on the right shows results from MALDI imaging analysis. The data shows that there is a presence of AMTRPs and AMASTs within the Africanized honeybee. However, these are not present in the non-aggressive bees. The figure on the left shows the imaging of mature AMTRP neuropeptides of aggressive and non-aggressive honeybees. Section A and B show the brain waves of non-aggressive and aggressive bees with AMTRP 88 through 96 highlighted in color. Sections C and D show the brain waves of non-aggressive and aggressive bees with AMTRP 254 to 262 highlighted in color. As you can see, the aggressive individuals showed brain waves that had more of the AMTRP neuropeptide than that of the non-aggressive individuals. The next set of data, the figure on the left, shows the imaging of mature AMAST neuropeptides of aggressive and non-aggressive honeybees. Section A and B show the brainwaves of non-aggressive and aggressive bees with AMAST 59 through 76 highlighted in color. Section C and D show the brainwaves of non-aggressive and aggressive bees with AMAST 69 through 76 highlighted in color. As you can see, the neuropeptides are mainly present in aggressive individuals. The research paper studied used spectral imaging to find that two types of neuropeptides called AMAST and AMTRP were linked to the aggressive behavior within the Africanized honeybees. So, why is this important? The research done highlights that we still do not know why or how these neuropeptides cause the aggressive behavior within Africanized honeybees, but it has been narrowed down to which ones cause the behavior. Future research should be done on these neuropeptides and what components create aggressive behavior in Apis mellifera. This is important for the study of other bee species and how these neuropeptides cause them to behave. The more research done on AMASTs and AMTRPs, the more we can understand about Apis mellifera. These are my references, and I have a link to my references page. Thank you for watching.